Hey guys, welcome back to Computational Biology with Python. In this section, we're going to move on from BioPython and we're going to look at the potential of using OpenCV, which is Open Computer Vision, which is another package kind of like the BioPython package. And we're going to look at it for the potential use in phenomics. And so what is phenomics? Well, we've had this exponential growth in our ability to sequence a genome. When I was in high school, the Human Genome Project was going on, which dates me, I know. But the cost for doing the Human Genome Project to sequence the first human genome was billions of dollars. And now you can sequence a genome for less than $1,000, and I saw Illumina, who is a company that makes these sequencers, say they're releasing a machine that can do $100 genome, which is incredible considering it was billions of dollars before, and now we're almost to $100. So that being said, we're producing tremendous amounts of genomic data, sequence data. The hang-up then becomes, well, knowing what a gene is great, but knowing what you want to associate what that gene is with what that gene does, right? So you have to marry genomics, the ability to look at genes and identify genes and sequence genes, with phenomics, their associated function, right? And that's kind of a big hang-up. So why am I interested in this, and why am I teaching this to you? So, well, I look a hot mess. All right. So I've had two positions. I've had multiple positions, but I've had two positions recently as a researcher. Right out of my graduate school, I worked for a really smart researcher named Ben Blackman at the University of Virginia, who's now at Cal Berkeley. And part of that project was to identify what genes control heliotropism. So heliotropism is kind of like a modified phototropism, or a plant, specifically a sunflower in this case, follows the sun across the sky throughout the day. Then at night, when there's no external cues like the sun, like light, it knows to go back to the east and wait for the sun to rise again, and then it does it again the next day. So I have a picture of this. You can see the sunflower following the sun across the sky, and then you get some shaking from the wind and such. And then when it turns nighttime, it goes back to the east without any external cue to wait for the sun the next day, which is a really cool problem. Or really cool feature, I should say, not really a problem. So that project required a genetic or genomic component, which was to use a GWAS panel, so genome-wide association studies. So it's a way to weed out what genes are associated with what physical traits, right? But in order to do that, we had to quantify these physical traits because it's a quantitative genetic technique. And so what it required was taking videos like this and then measuring every frame what the angle is so we could quantify that angle. Pretty labor intensive, right? After that project, I had, long story short, my wife got into a graduate school and I wanted to stay close to my wife, obviously. And so I followed her to the University of Mississippi, where I worked for Dr. John Kiss, and I worked on the spaceflight stuff. That's where I still do the spaceflight stuff today, the space biology. But part of that is we did this experiment on the space station where we grew Arabidopsis thaliana, and we removed gravity stimuli to look at just light stimuli, because light and gravity kind of work together in plants. So we wanted to just look at light stimuli. So we put these plants in the space station in these little tiny cassettes. They're about the size of a Matchbox car. And as you can see, excuse me, you can see their roots are growing in these different directions, right? Well, at the time when I did this, I wasn't quite the Python coder, and there are some additional issues I'll bring up throughout the course with this study, but I had to measure manually each angle of these root tips. There were 10,000 photos, I believe, or at least 10,000 cassettes, 10,000 of these images. So we had 80 some cassettes, and we had 30-minute intervals, and we had they ran for 2 days, and we had 3 genotypes, and we had 3 conditions, we had 2 light conditions, and then we had 5 gravity levels, and so you can see those all multiply together, right? So there's tons of pictures. So that the amount of time it took us to do the RNA-seq for this vastly paled in comparison to the time it took to actually measure the phenotypes, so we could associate the RNA-seq with the phenotype. Does that make sense? So that's where computer vision comes in. So computer vision is a way to process images with Python to ideally write a script to do these measurements for you, and then just loop through it and get all those things measured and put into a spreadsheet, and then you don't spend 6 months or whatever I spent measuring these things, right? So that's the whole point behind computer vision. Phenomics is going to be, it used to be that genomics was a big hang up because it was cost prohibitive. Not everyone could afford to do it, and that was mainly the reason. It wasn't an access to everyone. Now they even have sequencers that you can buy for, $5,000 that you can have in your lab, and they're the size of your cell phone. And so, but again, the problem becomes adding phenotype to that data. So let's start with some computer vision stuff here. So I'm going to make this a little bit bigger for you. Does that look good? Yep. Okay. So we're going to start with OpenCV. So we're going to import NumPy as NP, and then import matplotlib.pyplot as PLT for plot. And then we're going to type in matplotlib inline. Okay. Oh, I'm going to save this file. Save notebook as, we'll call this one, what did I call it over here? Okay. OpenCV basics dot my Python notebook. There we go. All right. So let's import our CV library. So we're going to import CV2. And we're going to have to install it first. So bear with me. Let's see, while this is loading real quick. While this is loading, we're going to be working with pictures today. So I've recently started a new project where I'm going to work with trying to grow mushrooms in space with a collaborator. So find a good mushroom picture, or no, find a good picture of anything you want. I'm finding a good mushroom picture. You can do, the example they used a puppy, but we're going to make sure that it's not this WebP file. Make sure you find a JPEG. JPEG, here we go. So I'm going to do these mushrooms. I'm just going to say mushroom. Okay. And now that we're back in practice, we're going to do... Okay, that took a little time, but let's look at how we install. So first thing we need to do is pip, what was the command? So we need to pip install upgrade pip because our pip version isn't the most up to date. And for you to update, for me, it says I already did it. And then we want to pip install opencv python. And it should install it for you. For me, it says already satisfied. Okay, so now when you write this import cv2, we should have it run. Perfect. We're going to say image equals cv2.imread, and we're going to say whatever your file was. So mine was mushroom.jpg. And then we can do type image, and it'll tell us it's a NumPy array, which is exactly what we want. So we can also, let's load it wrong, cv2.imageRead, and we're gonna say, I'm gonna copy what I have written here from the book, something, just a path that doesn't exist. IMRead, not IMGRead. So you'll see here, it still runs. It looks like it runs, right? And so like, oh, that's great, we found our image. But if you type image wrong, it'll say that it's a none type. That means that we've installed the wrong, or we've loaded an incorrect image, or an image in the wrong spot. So this is important to check to make sure your stuff is going to work. So let's do plot imshow image. And as you can see, we have our picture, but if you've noticed, my mushrooms have turned blue. So this is because matplotlib, which is the package that we're using for this, expects a different order of colors. So this is an RGB image, red, green, blue. Right, so it plots in that order, red, green, blue. But OpenCV expects blue, green, red. So we need to transform it. So we can say fix image equals cv2 color, or convert color, make sure you capitalize that C, image cv2 color BGR to RGB. Let's put an R here. Okay, and now if we do plot imshow fix image, I keep saying imshow. You see that we fix it, right? So that's kind of a nuance with OpenCV. You can also do make gray images. cv2.2.imread mushroom.jpg cv2.imread grayscale, and then in the same one I'm just going to do image gray dot shape, so we can see the size of our image in pixels. We're going to plot imshow image gray. Oops. Oh, there we go. So we said, oh, this is going to be our grayscale image, right? Well, it's a problem again with how CV2 looks at RGB images. So we need to transform it again using CMap. So we do plot imshow image gray CMap equals gray. There we go. So now we have a grayscale image. So you might want to, grayscale images have less information in them. So if you were doing something computationally heavy, you could convert to grayscale. We can resize our images. So fix image shape. So our fixed image was, so you can see here, this is the number of pixels, right? So 433 by 660. We can, the width ratio equals 0.5. The height, oh wait, I'm skipping ahead. New image equals cv2 dot resize fix image 1400, and then we're gonna plot and show new image. So you can see we've squished our mushrooms down because we've stretched it out really, and we've made this axis smaller as well. So if we do new image, or new underscore image shape, you can see we're 400 by 1000 instead of what were we up here, 433 by 660. We can do W ratio equals 0.5. Height ratio equals 0.5. New image equals cv2 dot resize fix image 0, 0. Fix image W ratio H ratio. And then we can do plot im show new image. Whoops. Oh, what happened there? Made it much smaller. Oh, that's supposed to be plot dot im show. Not image show. Im show new image. So while it looks the same as this, right? So we have this picture and this picture seem like the same size. Notice the scale, right? So this one, if we do new image dot shape, you can see it's 216 by 330, so it's cut in half. So we've used this ratio of 0.5 to cut our image size down in half. So let's do, we can flip images. So if we do flip image equals cv2 dot flip fix image 0, and then plot im show flip image. You can see we flipped it vertically. Now if we do flip image 2, cv2 flip fix image -1, plot im show flip image 2, you can see it is not only flipped, so this one flipped it just on the vertical axis, right? So it flipped this image down. This image is flipped down and backwards, right? Because you look at where this big mushroom is and it's on the opposite side. So you can manipulate that way. And let's end by saving an image tile. So we'll say type fix image to kind of see. This is a NumPy NDA or ND array or NumPy, I think NumPy. cv2.imWrite mushroom fixed image.jpg, and then fix image. It'll come back true saying it's right. So we look at our original mushroom, and then we look at our fixed, this was our fixed mushroom from above. Well, let's see, do we fix image? Let's look at this real quick. Let's change this. Let's do flip image, because that's one I remember. There we go. But you'll notice that it doesn't save the color convert. It only saves the image manipulation. Because we're opening it. So the reason is because this is opened in Jupyter Labs, which expects RGB, and then in Python, when we view it, it expects that other color. What was it? BGR. So now we're back to that same problem where it's blue. So you could use this convert command to go back to RGB if you wanted to write this and have it right in the correct color. And that's it. We're gonna stop there. So that's some basics. And I've actually had to use this because for my research installing cameras, there's a lot of times that the cameras, just based on the cameras I use, the way the wires come out, I have to have them upside down to take in the machines that I build. So flipping images is important because if I want to, turn it into a movie because I did a time lapse, or I want to present these figures, I need to be able to flip them. So it is important. It can be important, not necessarily like this is. So that is the end of this video. I hope you liked it. We're going to get more into the weeds with OpenCV and image manipulation. But if you're watching on YouTube, please hit like, please hit subscribe, and I will catch you in the next video.